Hi, I'm Aurora Pujol. I'm a geneticist working at IDIBELL in Barcelona, Spain. Our lab is interested in genetic disorders of the brain white matter. Today, I want to share our latest success story. It's about the power of clinical genomics and its impact on healthcare. It's about a global collaboration with patients from 4 continents to identify a novel ultra-rare disorder, and it's about disease modeling in zebrafish. Our study started with a collection of some 300 undiagnosed cases of leukodystrophies in Spain, for which we ran whole exome sequencing. We analyzed the data with our own algorithm, which uses human phenotype ontology terms. In one case, we spotted an homozygous loss-of-function variant on a gene not associated to disease previously, the DEGS1 gene of sphingolipid metabolism. Dysfunction of this pathway is found in neurological disorders such as Niemann-Pick, Dravet, or metachomatic leukodystrophy, so we had a good feeling to be on the right track. We contacted our collaborators Odile Busflug-Tanguy in Paris and Ali Fatemi in Baltimore, who run reference centers for leukodystrophies, as well as the NIH program for undiagnosed diseases, asking for second cases, and found a total of 18 additional patients.
Hi, I'm Dr. Ali Fatemi. I'm a pediatric neurologist at the Mosher Center for Leukodystrophy at the Kennedy Krieger Institute in Baltimore, Maryland, in the United States. We identified 2 individuals here that were siblings that had variants in DEGS1 gene, and through Gene Matcher we were able to identify the other individuals worldwide, and this collaboration started on that basis. This is a young child, a boy, who is having severe atrophy of his muscles. You see some nystagmus in his eyes. There is some facial weakness with a tented lip. You can see in his back the scoliosis and some hirsutism, and again the cachexia. When you look at his feet, there is scissoring of his legs, extensive posturing, spontaneously upgoing toes, and severe atrophy of the muscles.
My name is Marc Patterson. I'm a child neurologist at Mayo Clinic, part of the collaborative team who investigate and manage children and adults with suspected inborn errors of metabolism. The cases we're presenting today are a great example of our collaborative work. These were 2 brothers who presented to us at age 1 and 4 respectively with nystagmus and the subsequent evolution of a syndrome characterized by global developmental delay, hypotonia, limb spasticity, and varying degrees of dysarthria and dysphagia. Both boys were hypopigmented at birth, but showed gradual increase in pigmentation over time. They had dramatically abnormal MR images, which I will share with you now. This series of T2-weighted axial images was obtained from the older brother at 2 years and 2 months of age. On such images, mature myelin should appear hypointense, but you will see here at the arrow that normally myelinated structures are iso or hyperintense with grey matter, suggesting hypomyelination. We notice as well there is signal iso or hyperintensity throughout the temporal lobes, throughout the cerebral hemispheres. There's a little mature myelin occurring here in the basal ganglia and adjacent to the posterior limb of the internal capsule, but overall the picture is one of diffuse hypomyelination. 3.5 years later, observing corresponding T2-weighted axial images, we can appreciate that there are now dark areas of signal hypointensity in the white matter, but the appearance is patchy. This is present in the cerebellum, in the pons, as you can see here, with patchy hypointense or dark areas, and similarly, as we scroll up towards the cerebral hemispheres, we can again appreciate that there is now patchy signal hypointensity, that is the development of mature myelin, particularly in the posterior portions of the hemispheres, but now extending more generally. In addition, there is also an increase in myelination in the basal ganglia. These findings are quite consistent with a slowly progressive hypomyelinating leukodystrophy. Note also the ventricular dilatation and the fact that the cerebellum was small.
educate the candidate of Peking University First Hospital in China. My mentor, Professor Jimmy Wang, is pediatric neurogeneticist. The girl is one case of her leukodystrophy cohort. With the support of China Scholarship Council, I will learn whole exome sequencing data analysis in Professor Margit Master Lab in University of Michigan. We identified interesting variants in the DEGS1 gene, and then through the Meta platform, we connected Professor Aurora Pujol and offer in Spain, and participated in the study consortium. Thanks to the global collaboration, the patient has been diagnosed genetically and has access to specific drug treatment.
We then obtain fibroblasts from patients' biopsies and use a targeted lipidomics approach to quantify the substrate, the dihydroceramides, and the product, the ceramides, detecting 3 to 8-fold altered ratios in the patients. We found in the literature a drug in use for multiple sclerosis, fingolimod, who may inhibit the enzyme prior to the DEGS1, the ceramide syntase. We thought that accumulation of the dihydroceramides may exert toxic effects to the nervous system, and decreasing the substrate could ameliorate the patient's phenotype. As a proof of principle, we use zebrafish to model the disease and assay for fingolimod with very exciting results.
Clinics is a biotech company that uses zebrafish for understanding human disease and finding new treatments. We use a video tracking platform for measuring locomotion alterations, which is a powerful readout for getting insights into neural function. Here we see control, diseased, and diseased larvae with different concentrations of fingolimod. We see a restoration of neural activity in treated larvae. As this is a leukodystrophy, we have also looked at myelin-producing cells, the oligodendrocytes. Our results show a clear decrease of oligodendrocytes in diseased larvae and a restoration of the numbers in treated larvae.
We are grateful to our funding sources, as Laboratorio TV3, FIS, Fiberer, Speria Foundation. They made all this possible. We hope that this type of studies favor the use of exomes as a first-year test in neurogenetic disorders in Spain as well. Today, we are preparing a compassionate trial in these patients using fingolimod, which is a relatively safe drug. We are hopeful. Thank you for your attention.